It's my great pleasure to be here, and yeah, it's a very good opportunity for me to introduce the recent work. So my previous background actually is auditory neuroscience. I would like to briefly introduce why I got interested in this topic, because we know that in auditory neuroscience, time is quite important. For example, when you hear the speech I'm producing, of course, time is important. You need to extract the temporal structure from my speech, and your brain should follow the envelope of the speech signals I'm producing, and then you can process, and then you can understand what I'm talking about. And our previous work shows that not only that our brain signals could actually track the envelope of the speech signal, but also it's not just a passively following. It's kind of a doing temporal organization of the incoming auditory signals. So in other ways, our previous work showed that our brain actually segment and chunk the incoming auditory input in an active way to help us understand the auditory signals. So then I'm quite interested in this one is because I'm wondering whether we can find a similar role of time in vision. So how about vision? Can we find some critical role of time in vision? Of course, there are a lot of work showing that. For example, it's well established that neuronal oscillation mediates selective attention. Like Bob Desimo's lab, they have shown that gamma band synchronization between the high areas and low sensory areas, they can help to achieve the selective attention. For example, when the C and A, they have shown some gamma band synchronization, the stimuli at the A location will be selective attended and the neural responses will be enhanced. And also there are many, many labs showing that neuronal oscillations plays a critical role in different cognitive functions. And also we also know that our brain is not just a continuous processing thing. For example, the varying spiking probabilities will depend on the phase of the local field potential. So this suggests that our brain actually have some concatenate kind of windows of opportunity to come out. And so neuronal oscillation also reflect the rhythmic temporal cortex. And also, Rufang Van Rulen's lab in France lab, they show that they show that in both behavior and EEG studies, they found that actually attention not sample stimulus continuously, but rhythmically in 10 hertz. So these are backgrounds that showing that time is also quite important in vision, because previous people, when people are talking about vision, they are talking about space, shape, color, so not related to time. But many work show that time is also quite important in vision. So this is our study's motivation. Of course, selective attention is important. For example, when you drive a car on the road and the car in front of you, it's the most important thing you should mainly attend. But certainly there are some other things which is also quite important. For example, there are a dog maybe cross the road, and there are a passenger may also cross the road, and there are some incoming cars in the future. So you need to allocate in some way some attentional resources to other maybe tasking relevant items. But how brain does this? So we think attention must be efficiently and flexibly distributed over time and objects to accommodate different task contexts. And our hypothesis is temporal dimension might be important to organize and coordinate attentional allocations on multiple visual objects. So this is something I'm going to talking about today. We are interested in exploring the temporal structures in visual attention, and two parts. First, we want to see whether we can see dynamics in behavior in visual attention. In the second part, we use EEG to try to understand the neurodynamics in visual attention. So the first part is done by these two main persons. One is Kun Song. So he is my former graduate student. He's now at Max Planck for postdoc, and he's doing spatial attention. And also Dr. Yan Huang, he's doing like feature attention priming. But today I'm just talking about Kun's work. Okay, let's first see what's our traditional view about our attentional behavior. So we have behavior every day. But basically, we have a classical sluggish view for attention behavior. If you have watched this movie, you will know what I mean. So which means that although your neuron activities in your brain is quite fast, we believe that your behavior is not as fast as the neuron activity do. And the behavior is always sluggish and slow. So that's why when you look at the behavioral results in almost all the paper, for example, the y-axis is the behavior measurement like the reaction time or the performance accuracy, you will see the overall behavior performances like the bar. So you just compare the behaviors under different conditions and not think about time, right? Or you can, yes, measure the behavior as a function of time, but still we think about in a sluggish measurement. So you may see some slowly developed course in the behavior. But recently, there are some other views. So this is our previous perspective on the sluggish behavior, but now there is some other way. So maybe there are some hidden temporal dynamics in the behavior, and we can measure the behavior using a much finer temporal scale like this way. So using this way, from the behavior data, we can know whether we really see some faster dynamics. And there are some two recent work using this time-resolved measurement, and they got some interesting results. They found that actually behavior, yes, fluctuate as a function of SOA. So this means that our behavior is not as sluggish as we think before. So this we just use a very classical spatial pre-queuing paradigm. So I just briefly introduce you. Basically, the subjects need to attend to these two squares simultaneously, and then there will be a peripheral queue. Then after some time, the target will occur either at the queued side or unqueued side, and the subjects just discriminate the shape of the target and by pressing a key, and then we recorded their reaction time. So that's it. But the key parameters we manipulated here is the time between the queue and the target. We sample that in steps of 20 milliseconds, so which means we use 50 Hz sampling frequency to sample the behavior. Let's see what we get. So this is our results. And x-axis is a queue to target time, okay? And the y-axis is a normalized behavior measurements. And the different color just means different queue and unqueue, so two conditions. And this is a classical result. So people replicate it for many, many years. So people know this is right. But if you compare our results and previous results, you can see that they are different, although they share something. So the first thing we need to do is we need to do something and try to replicate the classical results. And what we find is that, very interestingly, if we just extract the slow trends of our measurements, we can replicate previous results. So this suggested that classical attentional effects actually can be replicated in the slow trends of our data. And this makes a lot of sense because classical measurements, they kind of are doing a sluggish measurement. That's why they are replicated in our slow trends. Then we treat our time-resolved behavior results. So remember, this is behavior results. We treated it as the brain signals recorded, like EEG, MEG. And we did a time frequency analysis of the behavior results. And this is what we found. So it's quite surprising that first, we find alpha pulses in the behavior time courses, and think this haven't been found before. In both, when the target either happened in queued location or unqueued location. So the x-axis is still time, the y-axis is the frequency, so you can see some alpha pulses in the 10 Hertz. And also, we find that there seems to be an alternating shape between the alpha pulses. So they jumps between these two locations. Remember, we have two square, so this means the alpha pulses will jump from the queued and unqueued, like alternative way. So we try to quantify the relationship between the queued location and unqueued locations. So we're just doing a subtraction. So valid minus invalid, and you can see clearly the blue, red, blue, red, blue. So this means the switching between the queued and unqueued locations in both left visual fields and right visual fields. Also, we noticed that the alpha pulses seems to be modulated by a much slower reason. If you look at this way, you can see that the interval between the blue and the interval between the red are all kind of around 300 milliseconds. So this suggested, and 300 milliseconds corresponding to a theta band region. So this indicates that the alpha pulse is modulated in a theta region. So we got such hypothesis. So the alpha pulse is fast, and it's happening in both queued and unqueued. And there seems to be a slow reason to modulate, kind of unseen hand to modulate the thing. And then we try to demonstrate whether there are some relationship between the alpha and the theta. And we just do a alpha theta power phase locking. Basically, the result shows yes. So there are a power and a phase locking relationship between the alpha and the theta in behavior results. And people know that previous electrophysiological recording, although show this in the electrophysiological data, but here we show that in behavior data. So this matches very well. And also this suggests a theta reason actually mediates the alpha band process in time. So let me summarize the thing we find in the behavioral oscillations. We call it behavioral oscillation because it behavioral results and it has shown some reason. So the typical attentional effects, it's in the slow frequency trend. And there seems to be a theta band reason. So the attention samples these two locations using a theta reason, like every 300 milliseconds. And within the location, we think it's an alpha pulse, so 10 hertz frequency. But what does this mean? The alpha band, 10 hertz, it's a kind of processing, or it means something. If you look at previous literature, especially EEG, actually alpha band is claimed to be inhibitory nature. So when people attend to the left visual field, people know that the contralateral alpha actually inhibit, not enhanced. So this suggested the alpha band doesn't mean processing, enhancing, it means attentional inhibition. So back to our results. This is a summary of our results. So we need to attend both of these two locations. And first, the queue locations. So it's queued, and then the attention will be captured there by producing an inhibitory alpha on the unqueued location. But attention will not be always there. They will shift to the unqueued location by producing inhibitory alpha on the attending location. And the reason is kind of a theta reason. And using this way, we can explain our behavioral results. Also, this we tried the similar thing on the central queuing paradigm. So now it's not peripheral queuing, but the subjects need to attend according to the central queue, and there will be 66% queue validity. And we just did the same thing, mirror the reaction time, and we find that slow trend can replicate classical results. And if you do the same analysis, we find very similar things. So this means that it's a kind of very general phenomenon. Okay. And also our results suggested that our behavioral results are not sluggish. It's not a flash in the Zootopia. It's a dynamic in nature. So the classical measurement just mirror the tips of the iceberg in the real behavior data. And also our results provide new perspective for behavior performances. Okay, I'm going to introduce the second bar, neurodynamics in visual attention. So this work is mainly done by Jia Jianrong. So he's my PhD student, co-supervised with Fang Fang. So here we want to use EEG to examine the neurodynamics. But one difficulty thing in EEG is that, for example, you need to attend to these two discs, but using EEG, it's difficult to dissociate the signal specific to the attended and unattended. So we use a technique which called the temporal response function. So what does this mean? So first I'm introduce the paradigm. It's quite simple. First there is an arrow cue telling you which side you should attend to. For example, here is to the left. And then there will be two discs have occurred, and you need to sustainably attend to like one disc, and because there will be a target randomly occurring there, and you just attend here 100%. But here, the luminance of the two discs, we actually modulate in time for 5 seconds according to two random and independent time series. So the luminance of this disc, they will just we just generate a random temporal sequence. So the luminance will modulate this way, and this way, this disk will modulate in another way, and they are independent. And we record the EEG signals. By doing a linear regression between the EEG signals and the time series, we can dissociate which we call temporal response function. So this actually represents a neuron impulse response, means that they can know from the same EEG signals, we can dissociate the response specific to the attended disk and specific to the unattended disk. And this is a kind of impulse response, which means that it's responses to each luminant change in the signals. And this method actually has been used in the auditory studies. So this is our results. We find that yes, you can successfully get a TRF responses, and this means that for each luminance increment, there will be a TRF. And when you shuffle the relationship between the signals and the sequence and signals, you cannot see the thing. And we, again, do a time frequency analysis of the results. We see the very prominent alpha band, but this is the onset. We still can see some sustained alpha band, but it's not very clear here. So then we can compare the TRF between the attended and unattended. So we do a subtraction between these two conditions. And again, we see an alpha inhibition. So this means when you compare the attend and unattend TRF, the attend shows the alpha inhibition in the first 200 milliseconds. And it happened in the parietal channels. So this is really replicate our previous behavior results. Remember, in the previous behavior results, we also find the initial alpha inhibition. And here we see the EEG signals still doing alpha inhibition. But also interestingly, we see a reversal trend here, although not significant. So in the following 200 milliseconds, there will be alpha enhancement trend. This also corresponds to our previous behavior results. So what does this mean? This means in the task, even I ask the subjects to 100 completely attend to this disk, but attention doesn't do this way. So they will first just attend to the disk as you required in the first 200 milliseconds by producing an inhibitory alpha on the unattended one. But like after approximately 200 milliseconds, attention will intrinsically jump to the unattended disk, although it's not task relevant, by producing inhibitory alpha on the attended one. So this is attentional switching. So it means that even when attention is instructed to focus on the attended disk completely, we still see the intrinsic sequential sampling of visual objects. Okay, so next question will be whether the spatial temporal attentional sampling pattern changes to a cardinal task context. So here we're using the same paradigm, but now we increase the Q target to 75%, which means that the target will happen at 75% probability on the Q disk, and also it has 25% probability to happen on the unattended one. And we just do the same analysis and see the TRF. And this is the result. We'll compare the 10 out of 10. You still can replicate the initial alpha inhibition, and you can see a very significant alpha enhancement reversal in the 200 milliseconds. So this suggested when the unattended disk, it's a little bit task relevant, the attentional switching just becomes much more prominent. We then decrease the Q validity more by decreasing to 50% Q probability. This means that target will be equally possible to occur at these two disks, and that's what we find. You can see that the alpha switching becomes much, much more robust and temporally prolonged. So let me summarize these results. This means that when you pay 100% probability to the A, your attention kind of doing a sequential sampling by creating a lot of attentional chunks. So you have A, and then you have a little bit like non-significant B trend here, like this way. But when your Q validity decreased to 70%, 75%, the B is the reversal trend becomes more prominent. And when they are both possible, you can see that the attentional switching becomes more prominent. Okay, our final question is that is the observed sequential temporal sampling specific to spatial attention? Because in the previous week, we just fixed the two locations. So it's maybe just focus on attention on space. But how about object space? So we do the same thing, but now it's different. So we ask the subjects to pay attention to the disk, but during the 5 seconds, the luminance will change, and also at the same time, they will run on the screen, like randomly. So their spatial location will not be fixed. And we did the same thing to extract the TRF and compare the attend and unattend. We see the very similar thing, alpha inhibition and the enhancement trend. So this is quite similar when they are not moving, they are there. So this suggested that the pattern we find is not specific to spatial attention, but could also be extended to object-based attention. Okay, let me summarize our results. So we show both behavioral evidence and neuronal evidence. So in behavior, we can see the behavioral oscillation and alpha pulses switches between these two locations in a theta region. And in the neuronal dynamics, we use TRF to examine the thing, and we also find that the attention seems to switch between these two locations by using inhibitory alpha pulses. So this suggested that attention samples multiple visual objects alternatively in time. So it's serial-like sampling. And the attentional spatial temporal profile adaptively changes to accommodate different task demands. When you change the Q validity, you can see the pattern actually changes. It also suggests that attention is dynamic and rhythmic. So this is a sequential sampling of multiple visual order objects. Go back to our original motivation. This suggested that, yes, you need to mainly focus on this car, but you also sequentially allocate attentional resources to other maybe task-irrelevant targets in some way. So, and also you will flexibly judge your attentional sampling profile to accommodate different contexts. Okay, so I already mentioned the behavioral oscillation part is mainly done by Song Kun. So yeah, and the EEG part is by Jia Jianrong. And I want to mention that my postdoc Liu Lin, he has using the TRF to trying to extract the neuron responses from global and local from the same recording. And he found that the global actually initiate fast in the parietal regions. And this is an MEG study. I think the data looks quite interesting, and welcome to her poster. And also want to thank my collaborators. So Fang, we collaborate on the EEG studies. Zhang Hang, my colleague, we are trying to create a computational modeling to model our results. And also, I want to mention that Meng Ming at Dartmouth College, we actually have some data showing that if we do fMRI, we can also see the oscillation in the fMRI signals using our same paradigm. Okay, thank you very much.